Imagine you've got a patient with a failing enlarged heart. We're talking about a condition called dilated cardiomyopathy.
Right.
It's where the heart muscle physically stretches out, it gets super thin, and it just struggles to maintain the pressure it needs to pump blood.
Yeah, it's a devastating structural failure.
Exactly. So genetic testing is done, and it points the finger at one highly specific culprit, a single gene called RBM20.
Mm-hmm.
Now, the logical next step in molecular biology seems pretty straightforward, right? You engineer a mouse model, you introduce a targeted delete or like a hard stop sequence to effectively just turn off that gene.
Right, you just knock it out entirely.
Yeah, you observe the structural fallout, and then you start developing a targeted therapy based on that.
It is the standard playbook for functional genomics, really. I mean,
Yeah.
you systematically disable a mechanism just to understand its baseline contribution to the whole system.
Right, but the twist in today's deep dive is that when researchers attempted to completely knock out this specific gene by putting a literal genetic stop sign right at its primary promoter, the biological system basically outsmarted them. They just, found a way around the roadblock and kept right on functioning.
Which is wild. And it forces us to reconsider a whole lot of assumptions about genetic resilience.
For sure.
We so often conceptualize genetics as this linear, static blueprint, right?
Mm-hmm.
But the data we're looking at today from Nature Communications, it reveals something much more akin to a dynamic, context-aware operating system.
Yeah, and so our mission today is to completely deconstruct this paper on RBM20. We're going to map out the hidden genetic architecture they uncovered, analyze the structural consequences on the cardiovascular system, and, look at how this completely changes the landscape of precision medicine.
It's a huge shift.
It really is.
Yeah.
Think of a gene as this massive, complex book.
Yeah.
We just assume the cellular machinery always starts reading at chapter 1, page 1. But today we're exploring what happens when the cell spontaneously decides to just skip the first chapter entirely.
Right. It starts reading at chapter 2 and ends up producing a functionally distinct story. And we are also going to look closely at the molecular triggers, the actual how and why behind these alternate starting lines, like how the research demonstrates how these hidden promoters are used to physically restructure the heart at the exact moment of birth.
Oh, wow.
And conversely, how the misregulation of those exact same pathways drives severe, totally opposite forms of heart failure.
Okay, so let's ground this in the primary function of RBM20 first. It codes for an RNA-binding motif protein, and its defining role in the heart is directing alternative splicing.
Right.
So for anyone listening who needs a quick visual, the transcription is like a camera capturing hours and hours of raw, sprawling footage. Alternative splicing is basically the director sitting in the editing bay.
That's a very apt analogy, actually. The director takes that raw pre-mRNA footage and decides which specific scenes the exons make the final cut and which non-coding segments, the introns, just end up on the cutting room floor.
So depending on how those exons are stitched together, that exact same raw footage can be assembled into a romantic comedy or like a psychological thriller.
Exactly. And RBM20 acts as the master splicing director, specifically localized right in the heart.
And the undisputed star of its editing bay is this protein called titin.
Oh, titin, yes.
Yeah, it's this colossal protein that functions as a literal molecular spring spanning the sarcomere in our heart muscle cells. It dictates the passive stiffness and the elasticity of the myocardial wall.
Right, and the structural demands on the heart, they change drastically over our lifespan. Like before birth, RBM20 orchestrates the splicing machinery to include highly elastic regions.
Okay.
So it creates this long, soft, compliant version of titin known as the N2BA isoform.
N2BA, got it.
But then, to handle the hemodynamic pressure of adult life, RBM20 shifts its editing strategy. It begins cutting out specific springy segments, particularly within what's called the PEVK region.
Oh, the PVK region, right.
Yeah, and that forces titin to become a short, rigid adult spring, the N2B isoform.
So it goes from soft N to BA to rigid N to B.
Right.
And it's not acting in isolation either. It's also regulating calcium handlers like CAMK2, making it a real central node for both structural integrity and contractility.
Absolutely.
Which totally explains the extreme lethality of its mutations. When we look at clinical data, specific mutations in the RS domain, that's the arginine-serine-rich region responsible for pulling the protein into the nucleus, or the E-rich glutamate domain, they consistently lead to familial dilated cardiomyopathy and sudden cardiac death.
Which is terrifying. And the therapeutic landscape right now is heavily focused on this exact dynamic.
Right, like what are the pharmaceutical companies doing?
Well, they're utilizing antisense oligonucleotides, ASOs, along with advanced base and prime editing techniques.
Oh, cutting edge stuff.
Very much so.
Yeah.
The clinical strategy for a condition like HFpEF, that's heart failure with preserved ejection fraction, where the heart becomes pathologically stiff, it often involves intentionally suppressing RBM20. The working hypothesis is that if you lower the functional dose of the splicing factor, it basically forces the heart to revert to producing those longer, softer, fetal N2BA titin springs.
Huh. Theoretically raising the adult stiffness.
Exactly.
But I mean, that introduces a pretty profound ethical and clinical calculation for the field. We're talking about intentionally shifting the architectural scaffolding of an adult heart back to a fetal state just by compliance.
Yeah, it's a massive intervention.
So for you listening, here's a question. If you could chemically loosen the springs in a failing, stiff heart, would you take that risk knowing it reverts the organ's basic structure?
It's a tough call, because our ability to safely execute that intervention relies on an incomplete understanding of how the gene actually operates under stress. Which, brings us directly to the experimental anomaly that sparked this entire Nature paper.
Yes. Let's get into the lax Z and knock and surprise. This is wild. So researchers set out to build a highly specific tracking model for RBM20. They took exon 1, the primary front door of the gene, and swapped it out for a lax Z reporter gene.
Which is a standard marker. It turns tissue blue when it's expressed so you can see it.
Right. But crucially, they followed it with a hard genetic estopopy codon, the sequence TGA.
So the design was meant to serve two purposes, right? Create a visual map of where the promoter is active using that blue stain, and simultaneously generate a null allele.
A complete knockout of the protein.
Exactly.
Because looking at the methodology, putting a TGA stop sequence at the very beginning of the transcript, that should terminate translation immediately.
Right. The ribosome hits that sequence, detaches, and you get absolutely no functional protein.
It should be a definitive molecular dead end.
That was the foundational assumption, for sure, and the initial histology supported it. The embryonic mouse hearts stained a deep, clear blue, confirming that the promoter was firing.
But the anomaly appeared when they ran a Western blot to verify the absence of the protein, right?
Yes. Instead of a blank lane on the blot, the assay detected a distinct protein band at approximately 180 kilodaltons.
Wait, so it survived the stop sign?
It did. The detected protein was slightly truncated compared to the canonical version, but it was highly abundant. And more importantly, functional assays showed that titin was still being actively spliced.
Oh.
The master director was still operating, despite the primary sequence being completely blocked.
Okay, let's do a quick recap and bullet summary for you listening before we get into how it did this. First, alternative splicing dictates how stiff or soft the heart muscle is. Second, RBM20 is the master regulator of this process. And third, scientists tried to knock it out, but they discovered the protein was still magically doing its job.
A perfect summary.
Okay, so if the primary entrance is physically barred by its stop codon, the cellular machinery had to have found an alternate route. How did they figure that out?
Well, the researchers utilized 5-foot race PCR and ribosome profiling RiboSeq to map this out.
Okay, unpack those acronyms for us.
Sure. So RiboSeq basically shows you exactly where the ribosomes are actively translating on the RNA. But 5-foot race E, that specifically isolates the 5-foot cap of the messenger RNA, proving the actual physical starting point of transcription.
So using those two tools allowed them to look beyond the standard coding regions and deep into the intronic space.
Exactly. And that is where they identified an independent transcription start site totally hidden within the first intron.
A completely stealth promoter.
Yeah, they designated this hidden door exon 1B, along with a slightly less active minor site they called exon 1A.
Exon 1B. So because the primary door, exon 1, was occupied by the TGA stop sign.
The RNA polymerase simply bypassed it. It just walked around the side of the house, initiated transcription at exon 1B, and the ribosome found an internal ATG start codon safely downstream inside exon 2.
That is incredible. And the resulting protein is structurally fascinating, right?
It really is. Exon 1 normally codes for proline-rich N-terminus.
The P-rich domain.
Right. The backdoor version synthesized from exon 1B entirely loses this P-rich domain. It's roughly 100 to 120 amino acids missing, depending on the mammalian species.
Okay. But it keeps something else.
Yes. It retains the leucine-rich segment encoded by exon 2, the L-rich domain. And crucially, the researchers used fluorescent tagging to watch this.
Oh, right. They tagged them different colors.
Green for the canonical front door isoform, red for the back door isoform, and they observed them perfectly colocalizing within the nuclear speckles.
Meaning they were hanging out in the exact same workspace inside the nucleus.
Exactly. Interacting seamlessly with core splicing factors like U2AF65.
So it's structurally truncated, but it's functionally competent.
Yes.
I mean, the implications of a secret start site bypassing a targeted knockout are massive. Which brings us to our second question for you listening. How many other knocked out genes in the history of medical research might actually have a secret back door we just never noticed?
It's a chilling thought for researchers. We have to assume the genome possesses layers of redundancy we are only just beginning to map out.
Yeah, I mean, decades of historical knockout studies across various fields might need a secondary review. If researchers relied solely on blocking a primary exon, they might have inadvertently been studying the effects of a backdoor isoform rather than a true null state.
Exactly. But the immediate question for this team was determining the physiological difference between a heart running on the primary canonical protein and one entirely dependent on this truncated backdoor isoform.
Right, so they isolated homozygous LAX-Z in mice, meaning models, surviving solely on this backdoor protein, and they ran detailed catheter-based hemodynamics and echocardiograms.
Right.
And the structural dimensions of the ventricles were actually within normal limits. But the hemodynamic data showed a distinct functional shift.
Yes, they saw increased deceleration time and a decreased early diastolic velocity, which is known as the E' wave.
E', right.
Yeah.
And additionally, the left ventricular end systolic pressure, the LVESP, was measurably reduced.
So looking at that pressure-volume relationship, we see a distinct mechanical profile.
Yeah, if you synthesize those specific metrics, a prolonged deceleration time and lower peak systolic pressure, it points directly to a highly compliant, softer left ventricle.
Yes.
The heart is physically more elastic. The backdoor isoform is clearly functional, but it appears to be slightly less efficient at processing its targets than the canonical version.
Right, and because its editing capacity is slightly diminished, a higher proportion of those long, elastic N2BA titin springs remain intact in the adult muscle.
It just doesn't cut them down as efficiently.
Exactly, and we see that gradient of sensitivity at the molecular level too. The PTVK region of the titin transcript is incredibly sensitive to the concentration and efficiency of RBM20.
Okay.
Whereas the I-band region is somewhat more resistant. The backdoor isoform simply doesn't drive the transition to the stiff N2B state as aggressively.
So this raises a critical evolutionary question. Why engineer a secondary promoter just to produce a slightly less efficient protein?
Well, the answer lies in multi-omics profiling. By analyzing mRNA-seq data from mouse and rat models across their entire developmental timeline, researchers track the expression of both of these isoforms.
Over time.
Right. And total RDM20 expression spikes dramatically right around the time of birth. But the critical finding is that the ratio of the canonical front door isoform to the alternative backdoor isoform is tightly, almost rigidly constrained during the perinatal period.
Wow. So before birth, the ratio is highly variable. As adults, it also shows significant fluctuation based on environmental stressors. But at the precise window of birth, the biological system just locks that ratio in.
Exactly.
I like to think of the transition at birth like operating a manual transmission while merging onto a high-speed highway.
Okay, I like this.
So the fetal heart is a low-pressure pump, right? Circulating blood through the low-resistance pathway of the placenta.
Right.
But birth is the highway merge. The heart must instantaneously shift gears, closing fetal shunts and transforming into a high-pressure, rigid pump capable of pushing blood through a massive, air-filled pulmonary system.
That's a great way to visualize it.
That tightly constrained isoform ratio is basically the biological clutch engaging perfectly to transition the tissue architecture.
And a disruption in that highly orchestrated developmental splicing program during the perinatal window, well, it sets a latent foundation for pathology later in life.
Yeah. Let's do another quick 5-minute recap for you listening. First, the backdoor isoform creates a softer, more compliant heart muscle. Second, the ratio between the two isoforms is strictly managed during birth to transition to an adult pump.
And this brings us to question 3 for our listeners. Do you think manipulating this birth switch in an adult could reverse the stiffness of an aging heart?
That is the million-dollar question right there, because it leads us to the most clinically actionable part of this data. What happens when this carefully balanced ratio goes completely rogue in human disease?
It gets really interesting here. The researchers examined two severe morphologically opposite conditions.
Right. Hypertrophic cardiomyopathy, or HCM, which is characterized by an overgrown, thickened, overworked heart muscle. And dilated cardiomyopathy, DCM, which is a stretched, thin, and floppy heart muscle.
Totally opposite structures. So to map the transcriptomic landscape of HCM, the thickened state, they utilized spontaneously hypertensive rats and validated those findings against a cohort of 97 human HCM patients.
Okay, and what did the data show?
The data showed that total RBM20 expression is significantly elevated in this disease state.
But the composition of that elevation completely upends the traditional model of genetic regulation, doesn't it?
No, entirely. The primary canonical isoform, the front door, is utterly unchanged from baseline.
Wait, really?
Yes, the entire pathological spike in RBM20 in these thickened hearts is driven almost exclusively by the alternative backdoor isoform.
That is crazy. And conversely, in the stretched, dilated heart of DCM, an analysis of 92 human patients again showed elevated total RBM20. But in this morphology, both isoforms rise simultaneously.
Right, with the canonical front door isoform heavily dominating the expression profile and driving the severity of the disease.
So we have two opposing structural pathologies hijacking the exact same genetic locus, but utilizing completely different entry points.
And the data shows that the differing entry points dictate entirely distinct downstream targets.
Explain that.
Okay, so in the thickened HCM hearts driven by the alternative backdoor isoform, the splicing changes are overwhelmingly clustered around actin binding, cell-cell junction integrity, and focal adhesions.
Meaning the spliceosome is prioritizing the structural scaffolding and mechanotransduction pathways of the cell.
Exactly, which perfectly aligns with the needs of a hypertrophic muscle desperately trying to structurally reinforce itself against high resistance.
Right, it's building thicker walls. Meanwhile, in the floppy DCM hearts driven by the canonical isoform, the splicing alterations target the contractile fibers themselves.
Yes, as well as the broader RNA splicing machinery, leading to just a systemic breakdown in contractility.
Interestingly, titin splicing remained relatively stable in both adult disease cohorts.
Yeah, that's because adult titin is already almost entirely shifted to its maximally stiff N-to-B state. The pathology has moved beyond the spring at that point.
So let's quickly recap this section. In thick hearts, HCM, the alternative backdoor isoform spikes. In stretched hearts, DCM, the canonical front door isoform dominates.
Which brings up question 4 for you out there. If two opposite heart diseases hijack the exact same gene but use different starting lines, shouldn't our heart failure drugs be just as specific?
Absolutely. The divergence in target preference between the two isoforms presents a huge challenge for generalized therapeutics.
Right. If two distinct disease states utilize different promoters of the same gene, a blunt suppression strategy is just insufficient. You can't just nuke the gene.
It requires precision targeting. But to build a targeted drug, you need to know what is actually pulling the levers in the nucleus. I mean, the DNA sequence for the RBM20 gene is identical in both the HCM and DCM cells.
Right. The DNA hasn't changed.
So how does the cellular machinery actually know which door to open?
Well, the researchers deployed JASPAR motif analysis to identify DNA binding patterns, and they used ChIP-TSAC specifically targeting H3K4-AME3 histone modifications.
Okay, H3K4-AME3. What does that actually do?
Think of H3K4-AME3 as a biochemical flag. It essentially acts as a chemical marker planted directly on the chromatin, physically marking DNA regions that are open, active, and functioning as promoters.
Okay, so it flags the active doors.
Right. And by mapping these active flagged regions against the known binding motifs, they isolated the specific transcription factors mediating the choice.
I love visualizing this. We can think of these transcription factors as highly specialized keys floating in the nucleoplasm, scanning for their exact matching lock on the chromatin.
That's a great way to put it. The binding of a specific key complex dictates which promoter activates.
So what were the keys for HCM?
In the thickened HCM hearts, they identified a heavy upregulation of a transcription factor called HAN-D2.
HAN-D2.
Yeah, combined with a downregulation of another called KLF7. This specific stoichiometric ratio creates a chemical environment that overwhelmingly favors opening the alternative backdoor promoter at exon 1B.
And the involvement of HAN-D2 is particularly telling, isn't it?
Very. HAN-D2 is a fundamental regulator of early embryonic heart morphogenesis. Its reactivation in adult tissue suggests the cell is attempting to deploy a developmental survival program resulting in severe structural remodeling.
It's trying to go back to the womb to survive.
Mm-hmm.
But in the dilated DCM hearts, the key ring is entirely different.
Oh, completely different. Transcription factors such as ZB1, SP1, and MED1 are heavily induced. And this specific combination acts as a highly potent magnet for the canonical front door promoter.
And the predisposition to this specific pathway is also hardwired into the genome of certain patients, right?
Yes. A genomic analysis of the DCM cohort revealed a high frequency of single nucleotide polymorphisms, SNPs, localized directly within the canonical promoter sequence.
So these are micromutations.
Right, and they physically alter the shape of the lock, significantly increasing the binding affinity for those specific transcription factors. It creates a genetic compounding effect that just drives canonical expression into overdrive.
Wow. So here's our final quick recap. Transcription factors like HAN2 or ZEV1 act as specific keys for different genetic doors. And precision medicine must target the exact ratio of these isoforms, not just silence a gene entirely.
And question 5 for you. Could the future of medicine be treating the keys rather than the genes themselves?
That is profound, because this molecular deep dive fundamentally alters the trajectory of cardiovascular pharmacology. As we mentioned earlier, the current clinical momentum heavily favors technologies like ASOs and CRISPR prime editing to universally knock down RBM20.
Right, just turn it off.
But this data clearly demonstrates that indiscriminate suppression could be catastrophic. You might temporarily resolve the contractile deficit by altering titin.
But you'd simultaneously destroy the alternative isoform's ability to maintain the structural scaffolding of the actin cytoskeleton.
Exactly. So the clinical target must evolve from total gene expression to isoform composition. We have to manage the ratio.
Modulating the transacting factors, the transcription factor keys, or physically blocking specific cis-regulatory elements to shift promoter preference, that offers a vastly more sophisticated and potentially much safer intervention than just silencing the entire genetic locus.
Oh, it represents a definitive shift in how we approach precision medicine. We are moving away from a binary model where a gene is either on or off toward a highly nuanced model where the specific ratio of isoforms dictates human health.
All about balance.
The genome operates with incredible contextual awareness. It assesses the physiological environment, whether the organism is navigating the perinatal transition, battling hypertension, or suffering from a dilated ventricle, and it physically shifts its transcriptional starting lines to rapidly adapt the architecture of the body.
It's brilliantly complex. And the complexity of that adaptation leaves us with a compelling final thought.
Lay it on us.
Well, we've seen how the adult heart retains a latent genetic memory of its fetal elasticity, right? Locked away behind a stealth promoter, just waiting for a specific combination of transcription factors to turn the key. It forces us to ask what other dormant physiological capabilities are hidden within our other vital organs.
Oh, wow.
Right, what latent regenerative or functional programs might exist in our neurobiology or hepatic systems or our immune response, simply waiting for the correct transcriptomic key to engage a hidden door in our DNA.
That completely recontextualizes our understanding of our own biological potential. The instruction manual is just far denser than we ever anticipated.
It really is.
Well, thank you for joining us on this deep dive into the hidden architecture of RBM20. We will see you next time.